Hello students, welcome to EPG Pathsala. I am Dr. Amarjyoti from Daulat Ram College, University of Delhi. In this module, today I am going to discuss cellular and molecular mechanism involved in maintenance of genetic information. After watching this module, you will have a fair understanding about various molecular mechanisms in maintaining homogeneity of genetic information. The learning objective of this module is to first understand the significance of maintaining genetic information. Second, how fidelity of replication is responsible for maintaining genetic information. Third, cells are exposed to several kinds of mutations during their lifetime. Here I will briefly discuss different types of spontaneous mutations resulting from environmental factors like ionizing radiations, oxidizing agents and metabolic products, as well as discuss the cellular mechanisms involved in countering spontaneous mutations and DNA breaks. Towards the end, I will review the consequence of alterations of genetic information. Discovery of DNA structure by Watson, Crick in 1953 has provided an experimental evidence that DNA is a double helical structure and opened door for contemporary researchers. Michelson-Stahl in his seminal experiment in 1958 provided evidence that DNA replication is semi-conservative and daughter cells receive one parent strand from and other complementary strand after each round of replication, which supports the fact that daughter cells receive identical genetic information after each cell division. It is now unequivocally accepted that DNA is the genetic material and all the information for coding structural and functional proteins are stored in the unique coding sequence of 4 nucleotides. These nucleotides arranged in DNA strand in unique sequence to encode information for synthesis of RNA and proteins, which plays crucial role in performing various cellular functions. DNA basically consists of 2 polynucleotide chain held together by complementary base pairing of purine and pyrimidines. The unique property of adenine, specifically binding to thymine and cytosine pairing with guanosine, leads to a complementary strand synthesis during DNA replication. Replication is not a foolproof mechanism. It is often associated with errors in replication. Apart from replication, cells also incur mutations due to environmental factors. These mutations could alter genetic makeup and lead to a loss of genetic information. In this module, efforts will be made to understand how different proteins and cellular machinery come together to counter mutations and errors during DNA replication. Cell grows and divides multiple times during their entire lifespan and perform their ascribed functions. On an average, stem cell in intestine divides approximately 5,000 to million times, whereas somatic or differentiated cells divide approximately 50 to 60 times. In the preparation of cell division, each cell undergoes duplication of DNA during synthetic phase of cell cycle to ensure that both daughter cell receives identical genetic information. Any alteration in duplication of genetic information would have potential to alter functions of daughter cells. Hence, during synthetic phase, replication of DNA is considered to be extremely crucial for maintaining genetic information. Cells have also evolved unique cellular machinery of achieving extraordinary speed and accuracy during replication. DNA sequence is maintained and replicated with high fidelity with a mutation rate of less than 1 error for a billion of base pairs incorporated, which corresponds to approximately 3 nucleotides mismatch per cell division. Both prokaryotes and eukaryotic cells have optimized their cellular machinery for proofreading and repair to maintain a uniformity within genetic composition over several generations. However, many of the times all these mechanisms fail, then normal function of cell gets altered, which could lead to either apoptosis or cellular malignancies. Every cell division is preceded by synthesis of new complementary strand, which with the help of DNA polymerase. DNA polymerase is a highly processive enzyme and recruits nucleotides at 3' hydroxy end of synthesizing strand at a rapid rate. In one of the binding event, DNA polymerase can add 5,000 to million bases for synthesis of new strand. Adenine and cytosine complements with thymidine and guanosine. Hence, newly synthesized strand are complementary to old parent strand. DNA polymerase 5' to 3' polymerization site have impressive ability to select correct nucleotides as well as distinguish nucleotides from dideoxyribonucleotide for complementary base pairing during DNA replication. Polymerization by DNA polymerase at synthesis site is not 100% foolproof, as incorrect base pairs are incorporated frequently at a rate of 1 base pair for every 10⁵ base pairs polymerized at the newly synthesized strand. DNA polymerase, apart from polymerizing activity at 5' to 3' site, also possess 3' to 5' exonuclease activity to eliminate errors inserted during replication. Wrong tautomeric forms of nucleotides are often associated with incorrect base substitution, which leads to incorporation of mistakes during replication. Proofreading of DNA synthesis is mediated by 3' to 5' exonuclease activity. As the name indicates, exonucleases are capable of degrading DNA starting from the 3' end of the newly synthesized strand. This proofreading property gives the DNA polymerase a second chance to add the correct nucleotide. The mechanism of correction is shown in this slide. Insertion of an error at replication site reduces the ability of DNA polymerase to add a nucleotide adjacent to incorrectly base paired primer. Actually, mispaired DNA alters the geometry of DNA polymerase and reduces the rate of catalysis at the polymerization site and increases the rate of proofreading exonuclease activity. Errors are recognized and growing strands slide away from the DNA polymerization site to the exonuclease site. After the incorrect base pair being removed, the correctly paired primer template junction slides back into the DNA polymerase active site and DNA synthesis is restored. DNA polymerase exonuclease activity act as a live delete key, which helps in deleting the most recent errors. On an average, DNA polymerase inserts one error every 10⁵ nucleotide added with the proofreading by 3-5' exonuclease activity. The error rate goes to 1 in every 10⁷ base pair incorporated. Proofreading at this extent is still not tolerable, as it incorporates more than 300 errors per cell division, which is not acceptable for a dividing cell. As described earlier, proofreading ability of DNA polymerase can allow incorporation of one error per million base pairs, which is not acceptable for dividing cells. The errors that are missed during DNA replication are later corrected by single strand mismatch repair system, as shown in the figure. Involvement of DNA mismatch repair increases the accuracy of DNA replication by 2 to 3 orders of magnitude. This mismatch repair enzyme scans the newly replicated genome for mismatches and accurately repairs. If a mismatch is found, then enzyme would identify and excise the mismatched base from the new strand and rectify the error, and original sequence is restored. This figure is showing the identification and excision of mismatch repair in E. coli and mammalian cells. Newly synthesized strands are often non-methylated for some time. In E. coli, mutase first identify the mismatch nucleotides by non-methylated adenine groups and recruits mut-L ATPase, which facilitates recruitment of mut-H hemimethyl ATPase. mut-H creates a DNA incision by its intrinsic endonuclease activity of mut-H. Mut-H then cleaves at GATC sequence and removes the unmethylated newly synthesized strand, followed by NIC. Cellular exonuclease removes 500 to 2000 nucleotides, which later filled by DNA polymerase and later sealed by ligases. Eukaryotic and mammalian cells lack mut-H. The MSH and the MLH are eukaryotic homologs of mut-S and mut-L. Eukaryotes have multiple mut-S-like proteins with different specificity. Some are specific for simple mismatches, whereas another recognizes small insertion and deletions. MSH and MLH binds to mismatched bases and directly excise and create a break in the DNA strand. In the leading strand, break is present at the end of the growing end, and in the lagging strand, nick is present at either the end of Okazaki fragment, which was later filled by DNA polymerases. MSH and MLH interact with the sliding clamp component of the replisome and would thereby be recruited to the site of discontinuous DNA synthesis on the lagging strand. Interactions with the sliding clamp could also recruit mismatch repair proteins to the 3' end of the leading strand. The importance of mismatch repair is highlighted by the fact that mutation in MSH and MLH are responsible for a common type of inherited colon cancer called hereditary non-polyposis colorectal cancer. Mutation in human homologs of mut-S are also responsible for about half of the all HNPCC cases. Post-replication mismatch repair would increase the incorporation of error from 1 error in 1 million base pairs to 1 billion base pairs, which corresponds to 2 to 3 mutations per replication cycle. Cells were exposed to different kinds of environmental factors like radiations, chemical-based mutagens throughout their lifespan. DNA is a highly stable biomolecule for storage of genetic information. However, their complex organic molecules are susceptible to spontaneous changes like depurination and deamination under normal cellular conditions. Approximately 5,000 purine bases and 100 pyrimidine bases are lost every day in single human cell due to depurination and deamination. DNA bases are also succumbed to damage by encountering hydrolysis, cellular reactive metabolites, environmental chemicals, and UV radiations, and many more. Nucleotides present in the DNA molecule could undergo deamination, hydrolysis, methylation, depurination, or dimerization under the influence of environmental factors like radiations, chemicals, and several other factors. These alterations in the intact DNA would potentially increase the rate of generation of mutation. This slide identifies various cellular mechanisms involved in countering spontaneous mutation resulted from depurination, methylation, or environmental factors during entire lifespan of cell. Many cells sense such elevated level of DNA damage and activate critical repair system by delaying the cell cycle via checkpoint mechanisms to ensure that DNA damage should be repaired before cell divides. Double-stranded breaks are extremely serious as no intact template remains for the repair, and to curtail deleterious effects, cells often initiate apoptosis. Double-stranded DNA breaks can be repaired either by non-homologous end joining or homologous recombination. In excision repair, damaged nucleotides are excised and then resulting gap is later filled by DNA polymerase and sealed by DNA ligase. DNA is repaired by removal of damaged bases followed by resynthesis of excised region. Exposure of UV light leads to formation of cyclobutane ring and pyrimidine dimer. The formation of such dimers distorts the structure as well as block transcription and replication at this site. Light photoactivating enzymes are present which derive energy from visible light to break the cyclobutane ring structure and restore the normal structure. These mechanisms are present in E. coli, yeast, and some species of plants and animals. However, photoactivation is absent in placental mammals, including humans. DNA damage like pyrimidine dimers and alkylated guanine residues are repaired by direct reversal of the damage. Methylation of guanosine at O6 position could lead to formation of O6-methyl guanine, which if not rectified would form complementary base pair with thymine. Enzymes like O6-methyl guanine methyltransferase are present in both prokaryotes and eukaryotes, which transfer methyl group from O6-methyl guanine to cysteine residues and restores the original guanine to maintain genetic information. DNA base excision repair removes specific type of single damage bases like uridine, hypoxanthine, abnormal bases or bases damaged by oxidation and ionizing radiation. Uracil is normally an attribute of RNA. However, occasionally they get incorporated during DNA replication. Deamination of cytosine often leads to uracil formation, which if persists, then it would lead to a complementary base pairing and results into a mutagenic event. Deaminated nucleotides were first removed by DNA glycosylase activity. DNA glycosylase excises uracil and cleaves the bond linking the base uracil to the deoxyribose of the DNA backbone. This reaction yields free uracil and a apyridinic site, a sugar with no base attached. Under normal cellular conditions, AP sites are spontaneously generated and several thousand of purine bases are lost. These sites are repaired by AP endonucleases, which cleaves around AP site and results single nucleotide gap is later filled by DNA polymerase and ligase. Within nucleotide excision repair, damaged DNA is first recognized, which gets unwound by helicase activity. Oligonucleotide containing thymidine dimer is cleaved by endonuclease and removed from the main strand. The gap is then filled by DNA polymerase and sealed by ligase. In this pathway, large multi-enzyme complexes recognize a wide variety of lesions like pyrimidine dimers caused by sunlight that distort the structure of DNA and removes the damaged bases as part of oligonucleotide. This mechanism is capable of repairing damage throughout genome and involves in transcription coupled repair. Once a bulky lesion has been found, the phosphodiester backbone of abnormal strand is cleaved on both sides of the distortion and the oligonucleotide containing the lesion peeled away from the DNA double helix by a DNA helicase enzyme. This large gap is then filled by DNA polymerase and DNA ligase. TF2H binds DNA by helix opening protein XPA and SPD. TF2H has two functions. First, it melts the DNA around a lesion during nucleotide excision repair and also help to open DNA template during the process of gene transcription. Double-stranded breaks are extremely serious as no intact template remains for the repair, and to curtail deleterious effects, cells often initiate apoptosis. Two mechanisms exist to repair double-stranded breaks within cell. Non-homologous end joining is simple and straightforward method where broken ends are placed adjacent to each other and joined by a DNA ligase. It is associated with deletion or loss of one or more nucleotide. This often results in mutation and gross chromosomal rearrangement which persists and often alters the genetic information. This is a quick and dirty solution of repairing a double-stranded break and often observed in somatic cells. In a 70-year-old human, somatic cell might contain more than 2,000 non-homologous joining scars scattered in the genome. However, these scars are not observed in the telomeres due to presence of telomerase activity. Homologous end joining is more advanced and effective way to repair the double-strand breaks in a cell. Here, broken DNA strand, instead of joining their end, uses accurate information from sister chromatid and synthesize the deleted nucleotide resulted from accidental breaks. Hence, this process often results in no change in the genetic information. In this, homologous recombination requires formation of holiday junction. Holiday junction often requires set of recombination protein which recognizes areas of intact homologous DNA sequence between two chromosomes and bring them together. A DNA 4-base junction or holiday junction is formed while addition of homologous nucleotides in the gap. Holiday junction resolvases enzyme then terminate the recombination process and separate the connected strand. Homologous recombination completes by DNA ligase after sealing the breaks, and this process results in no change of genetic information. In eukaryotes, DNA is tightly bound associated with histone protein in a repetitive array of DNA protein particles called nucleosomes. DNA packaged into nucleosomes to create a chromatin fiber which protects DNA from nucleases and allow compact packing of DNA within chromosome. Nucleosome core particle consist of histone core that consist of two molecules each of H2A, H2B, H3 and H4. Around these structure, 147 base pairs of DNA wrapped around 1.67 times. A fifth linker histone H1 is bound to DNA as it enters each nucleosome core particle. This forms a chromatin subunit known as chromatosomes. The packing of DNA with histones yield a chromatin fiber approximately 10 nanometer beaded fiber. The chromatin is further condensed through coiling into 30 nanometer fiber containing about 6 nucleosomes per turn. Apart from structural support of histones within nucleosome contains genetic information about expressing set of genes. Apart from structural significance of histones, these histones undergo several types of covalent modification, and these modification can carry important information regarding fate of underlying DNA. Information encoded within histones is reinforced as well through histone modification enzyme in reader-writer complexes during replication. Histone methylase complexes specially recognizes N-terminal histone tails that has been previously methylated at the same site the methylase modifies. The reader-writer complexes hence reestablishes the parental pattern of histone modification which can be inherited to daughter chromosomes. The faithful duplication in the pattern of histone modification is associated with epigenetic inheritance, which deals with heritable changes in cellular phenotype without a change in the DNA sequence. All the ever mentioned measures help in maintaining genetic information and attenuating incorporation of mutation during and after replication process. Although cells efficiently deals with different type of mutation, still it fails to completely eliminate the possibility of incorporating mismatches. During synthetic phase, one error is incorporated for every 1 billion base pair copied by the cell, and many such spontaneous generated mutations get permanently incorporated in genome and passed on to the next round of replication. Since eukaryotes have 2 copies of the same gene, that the effect of such mutation remains hidden in heterozygous conditions. Importance of these point mutations very much depends upon the location of insertion, since a point mutation at crucial site in homozygous conditions could lead to life-threatening genetic disorders like cystic fibrosis or sickle cell anemia. Many of the mutations may be silent and could accumulate within population to cause genetic polymorphism. However, occasionally some mutation produces proteins which have competitive advantage. Such changes provide competitive advantage to cells, which results in genetic variation. Therefore, maintenance of genetic information is important, but at the same time, error in genetic information paves way to genetic variation, which over time results in evolution. Mutations are permanent inheritable alterations in base sequence of parent DNA. Although many of these changes are of no significance, however, over several generations these changes accumulate, and in several instances deleterious mutation at crucial site often alters cells' ability to grow indefinitely, which often results in transforming cells into cancerous cells. In summary, replication machinery ensures quality and allows incorporation of not more than 3 errors in replication of cellular genome. Fail-safe mechanisms for proofreading and repair of mutation exist to maintain genetic homogeneity. Cells also have dedicated molecular machinery exists to counter spontaneous mutation induced by chemical and radiations. As an unique adaptation, stem cells divide slowly to keep the genetic pool error-free. Usually, daughter cells proliferate and differentiate into specialized cell types. Cells possess 2 alleles of a single gene, which helps cells in intrinsic repair by homologous recombination, as well as protect from mutations in heterozygous conditions. Sporadic genetic alteration could be deleterious or of evolutionary significance. In conclusion, cells have evolved a robust, complex DNA replication machinery for faithful transmission of genetic information from parent to offspring. Both pre and post replication checks during duplication of DNA maintains genetic information to a great extent. Accuracy of incorporation of nucleotide within complementary strand reaches as low as 3 errors per genome replication. Information stored in DNA is also exposed to chemical radiation or environmental mutagens. To counter spontaneous mutation, cells employ DNA repair enzymes which continuously scans, identify as well as rectify mutation to keep a check on the genetic alteration in both eukaryotic as well as prokaryotic cells. As a fail-safe machinery, cells senses elevated level of DNA damage and delays the cell cycle to increase synthesis of repair enzymes and ensure that DNA is repaired before cell divides. Failures of accurate replication and maintenance of genetic information could lead to disastrous consequences like transformation of these cells into cancerous tissue or cell death. Most cells with elevated level of DNA damage increases both synthesis of repair enzymes as well as delays its progression in cell cycle. Variation in genetic importance have its own significance. Although several mechanisms of DNA repair and proofreading are in place, but the cell genome are far from static. Mutations and gene arrangements are needed to maintain genetic variation and contribute towards evolution. A fine balance between genetic maintenance and variation exists, which plays a critical role in the development and evolutions of species. Maintenance of genetic information has been discussed in this chapter by highlighting the mechanism involved in minimizing the frequency of errors incorporation during and post replication. Genetic variation could result from spontaneous mutation due to environmental, chemical, radiation or UV exposure. Briefly, the cellular mechanism involved in rectifying these errors is discussed. Understanding detailed molecular mechanism of generation and elimination of mutations are beyond the scope of this chapter. In conclusion, this chapter briefly explains the significance and known mechanism of maintaining genetic information. Thank you.